Welcome to our kickoff 2020 Summer Café Sci series. I'm Deborah Bronk. I'm the president and CEO of the Bigelow Laboratory for Ocean Sciences. So Café Sci is a popular series of talks that we normally give to about 1,500 neighbors and visitors each summer, right in the lab. But we're excited to be offering it virtually for the first time. As a nonprofit research institute, the discovery, solutions, and inspirations we create are made possible by our many, many generous supporters. And I want to start off today by thanking each of our donors, board members, and advisory board members for their steadfast support of our ocean mission. I'd also like to extend a special thanks to HM Payson. They are a Maine-based financial advisor that's again sponsoring the series of talks, as they had for a number of years. Many, many thanks, HM Payson. I also want to encourage you to please use the question and answer button to send in your questions throughout the talk today. I'll try to answer as many of them as I can at the end. You'll also notice that raise a hand in the chat button, but these features aren't available for this event. For those of you who are new to Bigelow, I want to start off by telling you a little bit about us. And then I'm going to tell you about a story about a science mystery that in my career as an oceanographer, I was part of solving. Okay, so what is Bigelow? We are world-class leaders in creative, cutting-edge research, and we advance bold science for our blue planet. The lab was started in 1974, and since then our staff and scientists have just transformed what was an independent nonprofit from a scrappy little startup to a world-renowned hub of ocean discovery. In 2012, we moved into a new 60,000 square foot platinum LEED certified building that I'm sitting in right now, located in East Booth Bay, Maine. We study the foundation of global ocean health. This means we work primarily with the microbes, the little microscopic plants and bacteria at the base of the ocean food web. We also work with organisms like kelp and corals that make up the foundation of ocean food webs and other ecosystems around the world. We empower exceptional scientists. The lab consists of about 100 staff, and half of those have PhDs. Each of our senior research scientists runs their own research laboratory and is responsible for bringing in most of their salary and the salary of their staff through contracts and grants. But they're attracted to Bigelow for the freedom that they have to pursue cutting edge research and because they have a say in really how the lab is governed. We provide resources to drive their discoveries. State-of-the-art science requires state-of-the-art resources, and we have what we call discovery centers that support our research as well as research all around the world. These discovery centers include the National Center for Marine Algae and Microbiota. This is really a national treasure that houses over 3,000 types of microbes, marine microbes primarily, but some freshwater from all around the world. The Center for Aquatic Cytometry is a world leader in the development of new approaches to view and study microbes one single cell at a time. It also serves as the foundation of the Single Cell Genomics Center. First of its kind, it enables researchers to determine the genetic potential, again, contained in each individual cell. They're amazing facilities. And finally, the Bigelow Analytical Services Facility. This can run samples for toxin analysis, heavy metals, other pollutants. It serves not only our own institute, but the state of Maine and the rest of the country. So the history of science has shown that some of the great discoveries happen at the intersections of scientific disciplines. At Bigelow, we have mathematicians next to chemists, next to oceanographers, next to biologists that study disease. And our investments occur through individual projects and through impact centers designed to address specific areas. We are also known for making a big impact. First and foremost, we make discoveries. Our science is revealing the life and processes that empower the entire ocean. And while the sea covers most of the earth, unfortunately, much of it still remains a mystery. What we learn is vital to understanding how our society can care for and benefit from the ocean and has given life to this planet and really continues to sustain it. We also found solutions, which is one of the things that really attracted me to Bigelow. More than 90% of marine life can't be seen without a microscope. and emerging technologies are only beginning to illuminate the magnitude of this resource. We are always searching for ways our science can create valuable tools, spur economic growth, and improve environmental health. The incredible promise of this ocean innovation is the source of our passion and our optimism. And we strive to be a source of inspiration in the world. The future of our ocean and our planet will be decided by the actions of people. Our immersive education programs can open minds and launch careers. Our artistic partnerships, science communications, and public events like Café Sci share the importance of what we're learning. And our engagement with business and government leaders fosters innovation and helps us to inform decisions. Science can reveal opportunities, but it must also inspire people to embrace them. Today, I'm going to try to inspire you to care about water quality. Now, when it comes to water quality, scientists can mean many different things. Is the water free of pollutants like oil, arsenic, mercury? Is the area free of invasive species? And for much of my own work as a scientist, it means having the right amount of nitrogen. All of my research is focused on nitrogen. It's an element that all organisms need, and I've studied how nitrogen is used and produced by microbes at the base of the ocean food web all around the world. Each of these stars represents a location of one of my 50+ research expeditions. One area where I've focused a lot of my research has been in the Gulf of Mexico, along the coast of Florida. My work in Florida is focused on one of the most pressing water quality issues, harmful algal blooms. Just like flowers in your garden, blooms occur in the ocean. These microscopic plants called phytoplankton can form dense accumulations. All these green, this whole green area are billions of cells. These are microscopic plants, and this is what we would call a bloom. There are basically two types of harmful algal blooms. One is caused by too many cells, and a second is caused by cells that produce toxins. So let's first talk about blooms that are caused by too many cells. So this is a picture of phytoplankton cells taken with a microscope. Now, to make a phytoplankton cell, you basically need three things in abundance. The first is you have to have carbon. This comes from carbon dioxide dissolved in the water. The second is you need nitrogen. The third is you need phosphorus. Now, when it comes to the coastal ocean, the amount of nitrogen is the substance that generally controls how many cells you can make. If you want to know what's causing an algal bloom, a really good place to start is to figure out where that nitrogen is coming from. Now, blooms can be harmful for a lot of reasons. So if you have a lot of nitrogen coming in and causing a large bloom, if you have too many of these cells, it will actually cut off the light to the bottom. So things like seagrasses can't grow anymore. When these billions of cells die, they can create additional problems. They decompose, and the bacteria in the water are the ones that are doing that decomposition. Now, bacteria are like us. They take up oxygen. So when these blooms die off, you can get massive areas of very low or no oxygen. And these can kill fish, crabs, and other marine organisms. Then there are blooms made up of cells that produce these toxins. And some examples are domoic acid, which causes amnesic shellfish poisoning, saxitoxin, which causes paralytic shellfish poisoning, and brevetoxin, which can cause neurotoxic shellfish poisoning. Now you will notice a theme here. Shellfish can be one of the vehicles where toxins can go into other organisms. And that's because shellfish filter water out of the cells so they can concentrate the toxin and then pass it on to anything they eat that eats it, like humans. So today I'm going to tell you a story about the harmful algal blooms along the Florida coast that unfortunately combine both types, too many cells and toxin production. So here you can see the cells are this kind of cloudy area, and there are billions of them. This is a picture of one of the massive algal blooms that are also called red tides that have been devastating coastal Florida and the Gulf of Mexico for many, many years. Now these blooms are caused by an organism called Carina brevis. This is a single cell phytoplankton known as a dinoflagellate. And the cell was named to honor Karen Steiniger, a wonderful researcher who taught us a lot about this organism. And I've always wanted to ask Karen how she feels about having such a destructive organism named after her. I'm not sure if that's how I would want to be memorialized, but it was done with great respect and affection. So Corinne brevis produces a brevetoxin, and it's a powerful neurotoxin that can devastate marine life and cause massive fish kills. Here's a dead manatee. It's killed hundreds of manatees every year. Oftentimes there are mass deaths of turtles, and of course, you know, the ubiquitous fish kills. But one of the things about these blooms is that there's a mystery. We couldn't figure out what was causing them and how and why they lasted for so long. Remember, I said in the coastal ocean, if you want to make a lot of cells, you need a lot of nitrogen. So one question for some of these blooms, not all, but many of these blooms, is where is all the nitrogen coming from? And I want to say up front that when I'm talking today, I'm trying to use general concepts instead of pouring a lot of data out to you. So not every bloom in Florida is going to follow the information that I'm telling, but many of them do. So if you want to know where all the nitrogen is coming from, you want to produce a budget. Just like we have a home budget where we follow where the money comes in and where it goes, you want to do the same thing with nitrogen. In this case, you want to know how big the different sources of nitrogen to a region are. Here's an example of a nitrogen budget from the Wakulla Spring Basin in Florida. I did not produce this budget, I just lifted it off the web to give you an idea, and the colors matched the other figure. So in this case, for example, septic systems produce 34% of the nitrogen loading to the system. Now this information can tell you, you want to lower nitrogen delivery to the ocean. These are the big pots that you might want to go after in terms of sources. Now in our study, we wanted to know how much nitrogen was tied up in cells in a given region. So say, for example, and this is just hypothetical, this region off of Tampa Bay. And then we wanted to determine how much nitrogen was coming in from all the different sources within that region. These can include things like sewage treatment plants, terrestrial runoff, fertilizer from agriculture, atmospheric deposition. Rivers and streams can transport nitrogen way from inland, as well as industry. So here's the problem conceptually. We knew that there was this much nitrogen tied up in Corynea cells, but we could account for a fair amount of the nitrogen from all these known sources, but we were missing a big fraction of the nitrogen, but we had an idea. On where this mystery nitrogen could come from. So this is a picture that I've actually shown on a past talk at Bigelow. This is a picture of my very first cruise in grad school. Here I am. This cruise was studying trichodesmium. Trichodesmium is an organism that's really fun to study because it's found primarily in the tropics. This is Doug Capone. He was on my PhD committee. We called him the king of the trichonauts, which is what we call Doug and his colleagues who studied trichodesmium. And this woman is Judy O'Neill, and I'm going to mention her again in a little bit. So maybe remember Judy. Okay, so tricho is a fascinating organism. It occurs in two forms. It occurs in puffs and tufts. Now these are colonial organisms that consist of thousands or even millions of cells. They're visible to the naked eye, so you can pick these little pom-poms out of the water, or the tufts tend to look just like eyelashes. And they're unique in that they can use nitrogen gas as a source of nitrogen. So 78% of the atmosphere is nitrogen gas. And even though we're breathing in nitrogen right now, our bodies can't use that nitrogen. We have to eat protein to get our nitrogen. Now the gas, nitrogen gas is in equilibrium in the atmosphere and the ocean. But the vast majority of organisms in the ocean can't use nitrogen gas either. They have to take up things like ammonium or amino acids dissolved in the water. The issue is that nitrogen gas is composed of two nitrogen atoms and they're triple bonded together. So it takes an enormous amount of energy to break that bond, and most organisms just can't do it, except tricho can. So using the enzyme nitrogenase, trichodesmium can take up nitrogen gas, and then when they are eaten or then they die, a lot of that nitrogen is released as other forms like amino acids or ammonium. So this is how new nitrogen can get into the ocean. And once it's there, other organisms can use it, like the red tide Karenia brevis, which was our hypothesis. We believe that the mystery of the actual missing nitrogen was nitrogen from trichodesmium. So who's the we? This is a picture of the group. We are old friends from graduate school and members of our lab group. So here's Judy again. This was probably 10 years after that last picture was taken. She is now a professor at the University of Maryland and is an expert on the organisms that eat trichodesmium. Here's Cindy Heil. Many of you might remember Cindy. She spent 8 years at Bigelow, but was hired down at Mote Marine Lab to head up their Karenia brevis program. Since Karen Steidiger is retired, I think Cindy is widely recognized as the world expert on Karenia. Incidentally, the fact that we've known each other and have gone on so many cruises together is why I always tell graduate students, don't ever be a jerk. Because what you want to accumulate is friends through science over time, because they will have expertise that you will need to address hypothesis you want to test. So we called ourselves .gom, the Daughters of Trichonauts in the Gulf of Mexico. And our hypothesis was that tricho was taking up lots of this nitrogen gas, releasing other forms of nitrogen into the water, and Karenia was taking up that nitrogen to power the bloom. We wrote a proposal and got funding from the National Science Foundation. And here we are on the first cruise in the Gulf. So here's Judy, here's Cindy, here I am. This is the big instrument that we send over the side of the ship on a cable to collect water in these big cylinders. And we brought that water on deck and we would do our experiments. Here's the lab setup. We went a little insane in how much gear we brought out. We were only out for a week, but we really went hard at this issue. And for all the taxpayers that supported this grant, you got your money's worth. We were so excited to have our own project, and there were so many different things we wanted to test. So here's some more shots of us setting. Here's I'm setting up an experiment. Here we're all taking it down. So after 6 research expeditions, we found that tricho did in fact take up nitrogen gas, and that they did release it as organic nitrogen and ammonia, and that Karenia brevis would take up these newly released forms. So did we solve the mystery? That would be a big no. The rates we were measuring just weren't big enough to make much of a difference. Yes, it was happening, but it was a relatively small thing. And believe me, we tried. We made all kinds of assumptions to try to beef up our numbers to see if there was any theoretical way we could get it high enough, and maybe if we had done the experiments different, but we couldn't. We were missing something. So, like good scientists, we came up with new hypotheses. We wrote another grant, and this time we went back out got funded with a proposal from the National Ocean and Atmospheric Administration, and went out with a bigger team. This time we included physical oceanography in the project, and also dead fish. So in terms of physical oceanography, we thought that water movement and currents could be important to sustaining these blooms. For example, you can get winds or currents that cause areas of convergence where Karenia cells could be concentrated. So they didn't all have to be made in that one spot. They could be moved in from other spots. So let's go back to our earlier example. We were trying to determine the amount of nitrogen in Karenia cells in a given area. I'm just using this as a theoretical example. And then all the sources of nitrogen into that area. Now, most of that nitrogen is coming from the land. But what if we're concentrating, if the cells are concentrated by water movement? The Karenia cells in that area could then be coming from a much broader area, but are being pushed together by the currents. And it turns out, in many cases, this is exactly what's happening. We also got lucky in that we hit both big tricho blooms on our cruises. So here's a bucket of tricho that we collected using a plankton net. It looks very much like sawdust on the surface. We also hit big blooms of Karenia. Now you're kind of feel a little guilty when you get really excited over massive harmful algal blooms, but we really needed it to happen to study this time. When we worked on the side of the Karenia blooms, we would take the ship into the bloom, sample as quickly as we could, and then hightail it offshore until we were out of it. Because when you're on these blooms, it can cause real serious respiratory problems. So this is where we tried out our hypothesis that dead fish could be an important ingredient in how these blooms get so big. It's also when we did, hands down, the grossest experiments I've ever done in my entire career. So my student, here's a picture of her on the left. This is Lynn Kilberg, Dr. Lynn Kilberg. She did her PhD with me back at Virginia Institute of Marine Science on harmful algal blooms. And this was a postdoc in the lab at the time, Rachel Sipler, who has a really prestigious candidate chair position in Memorial University. So the three of us decided to figure out how much nitrogen you could get from a dead fish over the course of a week. Now these are important numbers because these data would allow us to model different scenarios. Say if we had 10,000 fish killed in a given area and they floated on the surface for 2 days before they sank, how much nitrogen could they release into the water? Or if they passed through the region on a current and it took them 4 days before they got from point A to point B in our study region, how much could they release? Because we would want to count that nitrogen in our nitrogen budget. So these are some cute little fish that are dead, that we stuck in bottles and then we let them decompose. I don't have any pictures of the bigger fish that we had out to sea that we stuck into bottles, but they were really gross. So these bottles were left to float in these bins in the hot sun and flow through water so that we kept them at the same temperature that the ocean was for a week while we were doing all our other experiments. And every couple of days we would take some of the water out of the bottle, filter all the gunk out of it, and then we measured the amount and types of nitrogen that was released by the fish back at the lab. And yes, it smelled just as gross as you're imagining. In fact, the very last filtration, we brought all of our filtration rigs out onto the deck because there was no way we were going to be able to do it in the ship. So this is some data that we generated. This is the amount of nitrogen that was released in micromoles of nitrogen per liter per gram wet weight of fish. And these particular experiments were over 72 hours. And this is looking at production of ammonia, which just went up, 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 up. Incidentally, 3,000 micromoles of nitrogen per liter is a massive amount of nitrogen. The concentrations of ammonia we were measuring out in the water at any given time usually would be less than 1 micromolar. So this was an enormous amount of nitrogen. Then other forms of organic nitrogen, there was a delay. Apparently the fish had to decompose to a certain point before these other compounds started coming off of it. But then we saw again a lot of nitrogen being produced. So we added this, what we called fish goo, to bottles that contained Karenia in it. And then we measured chlorophyll. So chlorophyll is the green pigment that makes plants green in your yard. It's a proxy for cell number because it's easy to measure. So instead of counting each individual Karenia cell, you can measure the increase or decrease of chlorophyll and it can give you an idea of whether the cells are growing. So here we've got basically our control, which didn't grow much at all. And then if you added the fish goo, the cells went crazy and and grew very well. So here's what we think are happening with a lot of these blooms. It all starts with you've got too much nitrogen released coming off the land. This can cause a relatively small Karenia bloom. But when the winds concentrate the cells by blowing them together, you can get concentrations of toxin high enough to actually kill fish. And fish are basically bags of nitrogen. Once those fish start dying, they're starting to release lots of this nitrogen into the water that the Karenia can use, which then causes a huge Karenia bloom. And at that point, you don't need the winds anymore to concentrate it. If the numbers get high enough, the cells can produce enough toxin to kill fish, and then it's like a runaway train, which is why they're so hard to stop and they can last so long. So if you really want to make a dent into these things, one of the things you should look at is stop putting so much nitrogen into the water. So what do blooms cost Florida? And I just pulled some of these out of the literature. In 2007, $51 million in losses to restaurants and hotels. Another study showed from 2015 to the 2016 season, $3.3 million lost revenue to the shellfish aquaculture industry. 2018, $8 million lost to hotels on Sanibel and Captiva Island alone. In 2018, $3.7 million in lost property sales. in Captiva and Sanibel Islands. And the state spent $14.5 million in beach cleanup costs in 2018. So these can be incredibly expensive. Unfortunately, I don't have a good number on the whole state because we don't have good reports. They haven't been done in years, really trying to collect all of this information so we can decide how big it is of a national problem. This isn't just the US, this is this figure, and I apologize, I couldn't find a good version of it. These are harmful algal events by country from 1980 to 2015, and here's the United States near the top. Now notice that these are reported blooms. So it could be we have a lot of blooms more than other places, but in many, especially developing countries, they're probably not even being reported because they don't have monitoring programs that track them. Another thing about these blooms is that they're getting worse. So this is from a paper in 2017 that is showing pre-1972, the various blooms, a lot of these on the West Coast, to the present. And again, the present in this case is 2017. And you can see the massive numbers of blooms that are starting to happen in the Northeast. Looking at another example for the cost of these blooms, they can really be devastating to local economies. So on the West Coast, there was a $97 million in lost Dungeness crab landings and an estimated $40 million in lost tourism spending in 2015 alone. Now, closer to home, what is very concerning is that we're starting to see harmful algal blooms, including perennial species, in the Gulf of Maine. We're also seeing Pseudonychia in the Gulf of Maine, which is the species that causes the massive blooms on the West Coast. This is heartbreaking because Maine is some of the most pristine coastal ocean waters in the US. That's why it can be such a great natural laboratory. And the problem is that these issues are not getting a lot of attention. Since coming to Maine in 2018, I've gotten questions about harmful algal blooms when I give talks, and yet there are relatively few people in the state that work on these issues. By comparison, Virginia and Florida both have small armies of researchers working on these, and yet they're still losing the battle. So the main message I want you to take home with you today is that it is easier to prevent something than it is to clean it up. And I believe Maine needs to get ahead of these issues, and we are running out of time to do it. What we don't want is for Maine to look like Florida in 30 years. For that reason, as part of our new strategic plan, we're launching a new Water Health and Humans initiative. I want to model this initiative after our very successful Center for Seafood Solutions that I told you about earlier. The Director of Seafood Solutions, Nicole Price, partners with state officials and local industry to solve problems. So for example, the nascent kelp industry doesn't have the resources to fund research on best practices, but Nicole works with them to identify problems, and we raise the money through grants or philanthropy to do the research they need to solve those problems. It's a really unique but positive but powerful approach. I could envision this new initiative working with local towns or even homeowners, the Department of Environmental Protection in Maine, wastewater treatment plants in Maine, and the Department of Marine Resources. These are the kind of partnerships that can really bind researchers across the state together to develop solutions and really serve the taxpayers and the community well. Now here's one example of the type of work we'd like to do through the initiative. It turns out that the coronavirus that causes COVID-19, SARS-CoV-2, can be detected in effluent. So if you've got here, we've got waste coming from hospitals, urban areas, suburbs, farms. They enter wastewater treatment plants, and by monitoring those plants and the effluent and using the techniques that we use to study the ocean, we can detect COVID-19, SARS-CoV-2, sorry. And this is a great example of how we can use the skills we've acquired working with microbes to help solve a current pressing problem. A few weeks ago, we submitted a proposal to fund this work, and in drafting that proposal, we worked with the Maine CDC, Center for Disease Control, the Maine Department of Environmental Protection, and wastewater treatment plants around the state, because these are the people we would send the data to. What's important is that this could be a way to detect when the numbers of infections are increasing in the area without having to sample and test each individual person. So to end, I always like to give people ideas of what they could do to help. One thing you can do is don't use fertilizers. Your grass probably doesn't need it. And what you put on your grass or your garden will find its way into the ocean. Unless you are very skilled at measuring out and using the appropriate amount, it will wash off. Another is when you're building or involved in building projects, try to limit impervious surfaces. We want the rain to land on the ground and go into the ground. We don't want it running off and carrying nitrogen and other forms of material into the ocean. Support science. Support education programs that teach science. Support science organizations. You don't have to support Bigelow, though we'd love it if you did, but get involved in science organizations and promote it. This country is dealing with so many issues, and science, whether it's social science, economics, physical science, environmental science, will be key to having us solve them. And finally, vote. Vote for people that appreciate the value of science. Vote for people that want to use the best available information to address issues and problems. And that means people that will work with scientists to get that information. So to end, I want to say thank you to everyone that pays taxes, because you support a lot of the research that we do. I want to thank all of our donors. Without you, we could not do what we do. And I want to thank all of you for joining me this evening. I also want to invite you to call in or Zoom in next week, where the second of these Café Sci talks will be given by Barney Balch, who's a senior research scientist here at Bigelow. His talk is Distant but Connected, a journey to discover how Antarctic algae shape northern oceans. And with that, I would be happy to take any questions. Thanks for listening.
Great. Debbie, we do have a couple questions, and we do have time for some additional ones. So if anyone does have questions they'd like to ask, just use the Q&A button at the bottom of your screen to send those in, and we'll try to get to as many as we can. The first question we had was about Corinnia brevis in the Gulf of Maine. You know, is it a problem that has been around for a while? And if not, why not?
I don't know the answer to that, and I don't know if anybody does. So the species of Corinnia that's been found in the Gulf of Maine is an organism called Corinnia micki-mortoi. So it is a related species. How long has it been around? We don't know. Unless you're specifically looking for things, it's hard to tell. One of the ways that we're dealing with that issue, because you always ask yourself, is it new? Has this always been happening and we weren't paying attention? Is we've got a new project called Environmental DNA. And this is a partnership with the University of Maine and other labs across the state. And the idea behind environmental DNA is you can collect a water sample from out there, and then just kind of through brute force, you can isolate the DNA that's in there and determine the organisms where it came from. So you don't have to sit and look through a microscope, which is very labor intensive. And you have to have great knowledge about phytoplankton taxonomy to determine all the different types of organisms that are in there. This is all done using molecular tools, and it can give you a snapshot in that period of time what organisms were there. This is going to be an incredibly powerful management tool in the future, because you will be able to say, you know what, yep, it's there, it's there, it's there, it's there, it's not here. You can watch to see if the numbers increase. This will be valuable from the standpoint of fisheries management, for example, because you'll be able to know what fish species or right whales were they in a given area in the last 24 to 48 hours. Now, there's a lot of basic research that still needs to be done on this tool for us to really understand what the data is telling us. But this is a 5-year project funded by the National Science Foundation, and I think it's going to be fantastic for Maine. And if this work had been done 30 years ago, I would be able to answer that question right now.
Great. Well, we've got another question here about the effect of harmful algal blooms on the lobster industry in Maine.
Yeah, so I probably should have anticipated this question. I don't know how affected lobsters are from a toxin perspective, because they don't lobsters eat bigger things, all right? So they'll eat things that fall down onto the sediment, but they are not going to eat Karenia, for example. So they could be impacted if you were to get a really thick bloom of any species, talking about the too many cell type of harmful blooms, where you would get low oxygen areas, that would force them out of region. So it would impact it that way. But I do not believe they are impacted in the same way in terms of accumulating a lot of toxins, because that's not, they don't filter tons of tiny particles like Karenia out of the water. I hope David Fields is listening and he can confirm if I said that correctly.
Okay, next question here is, in addition to causing toxic algae blooms, phytoplankton serve as the base of the aquatic food web. Is there a way to achieve a balance so that toxic blooms do not occur, but that there are sufficient populations of other phytoplankton to support the food web?
Yeah, so phytoplankton will live wherever they live. What you want is you want the communities of phytoplankton that should be in that area. Now, like any place or like any organism, phytoplankton are adapted to certain environmental conditions. So some in the open ocean, if you go out in the open ocean, there's very, very little nitrogen. It's really kind of a nitrogen desert. So the organisms out there are adapted to living in conditions with very, very low nitrogen. They're still out there. They're doing quite well. They're happy. They're the ones that should be in that area. When we dump a lot of nitrogen into the coastal ocean, we are changing those environmental conditions, and we are selecting for kind of weed species that can grow really quick and take up, all of this nitrogen that actually, some of the phytoplankton species in the open ocean might actually find toxic, right? Because they're not adapted to that environment. So, so by trying to keep our environments as close to their natural state as possible, you will have organisms that tend to be better for the environment. Let me give you an example. So when I lived in Georgia, I bought a little cabin on a couple of acres. Now, there were beautiful pine forests everywhere, but on my 2 acres, they had basically cut down big areas and disturbed that environment. So what grew back was poison ivy, because that was one of those species that likes disturbed areas. And it shouldn't have been there, but it was everywhere. So in a sense, when we disturb an environment like the coastal ocean by putting lots of nitrogen in, or toxins, or any number of things, we will change that community. And the best thing we can do is try to put it back into its natural state. I hope I answered that. Yeah, I'm going to stick with that answer.
Great. We have a few different questions that are asking about different types of aquaculture and their effect on nitrogen release into the environment and what the concerns there might be.
So let's see, there's a lot of ways you could take this question. So one thing about aquaculture, shellfish filter a lot of material out of the water, and that can actually be a really good thing. So I did my PhD on Chesapeake Bay. The belief based on sediment data is that before the Europeans came to North America, there were so many oysters in Chesapeake Bay that they filtered all of the water in the bay like every 3 days. So oysters are an organism that naturally can clean the water. Now, when this paper was published when I was in grad school, there were so few oysters in the bay that it was like 375 days. So building up aquaculture, and especially with shellfish, can be a way of cleaning the bay. You're taking cells that might be too abundant from the water and cleaning it out of the water. Now, if those are non-toxic cells, that's a win-win. You're cleaning the water and you're producing a product that people love to eat. the important thing is you've got to monitor for those toxins. And one of the things Bigelow does is we monitor toxins. We work with the state so that if the toxins get too high, we can say, you know, don't catch shellfish right now. We got to wait until those cells, those shellfish clear themselves of the toxic species. Another, I'm not sure if the other way you could be going with this question is looking at, there's some land aquaculture. So there are large salmon farms. There's 2 large salmon farms that are going in. Both of those could really create some problems in terms of nitrogen if they don't deal with their waste, because fish make a lot of waste. And I know there's a lot of concerns. I've actually seen their plans, and the wastewater treatment plants they plan to use to treat their waste. And so, yes, aquaculture can cause problems if it's not done correctly. From what I have seen, I don't think that's the case with the 2 salmon farms. And if somebody wants to give me a more specific question about that, but those are the kind of 2 general areas that I think aquaculture can really impact nitrogen.
No, I think that's great, Debbie. And someone actually followed up with a question about what you just addressed. So I think you've covered it.
Okay.
Next question is about the effect of water temperature and how that might be a concern for this issue.
Yeah, so water temperature is tricky. So because water temperature does 2 things. If it warms up, the warmer the water, the faster cells usually grow. That's why if you're going to buy chicken and going to cook chicken, you know, tonight, you want it to have kept it in the refrigerator and kept it cold, because keeping it cold retards microbial activity. You wouldn't want it sitting out on your counter at room temperature all day, because the bad bacteria are going to grow much faster there. Same thing with microbes in the ocean. You warm up the water, you tend to warm up the rate of growth, increase the rate of growth. So warming, part of the reason why Corenia and cells in the Gulf of Mexico grow so fast is because it's so warm. So as we're looking at increasing temperatures in the Gulf of Maine, one expectation would be you're going to see an increase in microbial activity. That doesn't necessarily have to be bad, but that's something to think about. What can be more insidious is temperature also will affect water density. So there are 2 things that affect the density of water. One is the amount of salt, and one is the temperature. So if you've got fresh water coming into an area, and that fresh water is warm, that is going to be light water, relatively less dense water. And that can, and if that is, and it flows out over colder, denser seawater, it's not easy for those 2 water masses to mix, those 2 layers. And that can inhibit oxygen getting into the water. This is, I'm not familiar with too many issues of it in Maine, but it's a big issue in Chesapeake Bay. This lack of mixing, just like oil and water, it's 2 different layers, they're different densities, and you really have to shake up that bottle to get those 2 layers to mix. The same thing can happen. And so if you've got a bloom forming, things are dying, you're capping it off from the atmosphere, so you're inhibiting oxygen being brought back down into the water, you can cause areas of low oxygen much quicker.
Great. Next question up is that this person's heard about ocean acidification affecting shellfish and is wondering, does this impact the toxins and their filtering?
That's a really good question. I'm guessing it probably could. So when shellfish are in more acidic waters, you can think of it as they're basically getting an extra tax put on them. It just takes more work to continually maintain and build shell, because that shell is in a chemical environment that wants to break it down. So anything that causes an organism to expend more energy on something other than growth or getting food could have a negative impact. Yeah, so I'm guessing there could be an effect of ocean acidification, because they are having to work harder to do kind of what you think of kind of a background job of just maintaining their shell. Can you read that question again? I want to make sure I answered that, Stephen.
Yeah, I think you did. It's they just said, I've heard about ocean acidification affecting shellfish. Does this impact the toxins and their filtering?
Great. Okay, so if so, where it could impact it would be if they're spending more energy maintaining their shell, they might not be feeding as quickly. If they're not feeding as quickly, they're not going to. If they're in a toxic bloom, they're not going to accumulate as much toxin. But yeah, so that's a really good question. I'm guessing it probably wouldn't have that big of an effect, but it could, because like I said, it's kind of an extra tax on the organism.
Great. And I think we've got time for one more question. So that is, what are the chances of success with this issue? It seems like such a huge problem.
So what are the chances of success with harmful algal bloom? So in a place like Maine, I think there's great chance of success to, and that is because Maine doesn't have a huge population. Maine has so there is an ability to know and work with all the key people you need to address a given issue, for example. Whereas in a place like Florida, where there's so many people, there are so many state agencies, there are so many different scientists and industry people, you really, to try to change how things are being done, you have to convince and impact an enormous number of people. In a place like Maine, you don't have to impact that many people that are making decisions to change how things are done, which is why I think building relationships between resource managers and industry and scientists and DMR, the CDC, the governor's office. In a place like Maine, I am so hopeful that we can tackle these issues because, and really be a model of what it takes to do that, because we're kind of a little microcosm. We don't need to impact that many people compared to other states to really change how we're doing things. And that gives me an enormous amount of hope. The other thing is, because we have that potential, we can really be a test bed for some of these things. Not just harmful algal bloom issues, but lots of issues. Because one of the debates going on in a lot of states on science issues is there's so much, what's the right way to do it? Let's use the Gulf of Maine, let's use Maine coastal waters that are not as perturbed as other places to show other states the right way to do it, because we have such a unique opportunity.